source, and have vitamin gummies. We have a whole lot of combinations. Sometimes we have customers come up with their own combinations. So we have multivitamin gummies, and we have gummies for stress. Then we have gummies for sleep. So we have gummies for eye health, which is contains xanthan, etc. Caffeine. Then you have gummies for bone health. Then you have gummies for women, where you have iron, etc. And we also, like when you say gummies for stress, apart from probiotics, we also have as ashwagandha and other kinds of herbals, etc. So it all depend on the different conditions, and the challenging aspect for us to have probiotics in a gummy format, because when we manufacture gummies, it requires a lot of heat. I mean, it goes through the manufacturing process. So most of the probiotics which we use are the bacillus species, the bacillus coagulans, and we also have to coat them so that they're not destroyed during the manufacturing process. Yeah.
Ma'am, good afternoon, ma'am. Ma'am, is there any predominant probiotic prescribed for neuro-related or the brain-related symptoms, or the stress and I mean the things like that?
See, we developed a formulation which is a multi-strain probiotic formulation of Lactobacillus casei, reuteri, plantarum. We chose them based on their ability to produce GABA, and but then again, if you look in the, you go to Amazon and you say probiotics for stress, you will find lots of different strains. So it'll all depend on whether they've been clinically validated, and whether any kind of clinical trials have been done. Then the different companies, in fact, there's a company which only its product is only for mental health, and there the probiotic strain is plantarum. So it depends. It varies from study to study. But we will promote our Cognisol, that's a brand name, which is a multi-strain probiotic formulation. And some of the disadvantages of lactobacillus species is that most of them have to be refrigerated. So this is where, when you look at it commercially, cold chain storage, it has to be refrigerated, including in the pharmacies, etc. Customers should be willing to take a refrigerated product. So especially when you and in India especially, I do find that most of them are not stored at in the refrigerated condition, though they're supposed to be. So I really wonder if they are viable. They are not viable, honestly. So when you look at the label, some of them say three, like it's the strength of a probiotic is generally given as CFU. So it'll say CFU as at the at the time of manufacture. But what is important is that it should promise the strength till the expiry date. That's also very important. So there's a lack of awareness on how probiotics have to be stored, and especially because of the costs involved, it becomes difficult.
These normal probiotics which are prescribed, like the Sporlac and Econom, which contains Saccharomyces boulardii, is it to be stored at room temperature?
No, yeah, that can be stored at room temperature.
Thank you.
So this Sporolac is Bacillus coagulans. It's Lactobacillus sporogenes is an old name. Later it was changed to Bacillus coagulans, and now it's been changed to Weizmannia coagulans. And now there's another name which I myself I don't remember. So they've been changing the classification, the name of.
Ma'am, will they undergo the same clinical phase trials like all other drugs do, or it can be used directly? Or it will come into the market directly?
So we have a food license. Most of them are supplements. So we don't really have to go through the regulatory route of applying to the DCGI for permission, especially because they're considered as supplements. Unless we want to have some claims, clinical study, and then we have to take the approval of DCGI, and then we have to submit it for approval.
Okay, not even animal studies, they will go for preclinical studies.
We do clinical studies. Yes, animal studies can also be done. So based on a few animal studies, we also filed patent applications.
Hypersensitive reactions, note the use of
With our studies so far, none, but yes, with probiotics, there can be bloating, which is one very common side effect with probiotics. And if also it's very important that the person is not allergic to any of the ingredients, especially when we use excipients which contain lactose. They have to be, not allergic to lactose or gluten. So these kind of precautions are which we'll have to, take. Yeah.
Thank you, ma'am.
One second now. Thank you once again, ma'am, for addressing the questions. I kindly request Dr. K. Anuradha, ma'am, to honor Dr. Jayanti, ma'am, with a memento as a token of appreciation. Sai Padma, ma'am. Thank you, ma'am.
For the next session, as we have a small delay in the arrival of the speaker, we would like to invite our in-house speaker, Dr. S. Vanita, who has done her doctoral research work in the field of prebiotics, probiotics, and postbiotic, and the role of FOS on growth of lactobacillus species and production of short-chain fatty acids using gas chromatography. I welcome Dr. S. Vanita to present her deliberations about the research that she has carried out in our college.
Good afternoon to one and all. It's almost post-lunch time, I think so. Hope your gut microbe is doing some murmuring. I can hear it till here. So I'll take you to a small journey where a small part of my work, what I have done during my research, I would like to share some, results, what I have got, and they were very interesting also. So let me take you a small journey. And the topic I have given as, it is a journey starting from prebiotics, going towards probiotics, and landing up on postbiotics. So three topics we have joined together. And also it is indicating about the role of FOS, that is fructo oligosaccharide, on the growth of lactobacillus species, and SCFS stands for short chain fatty acids production using gas chromatography. So since you all know me, I'm just going ahead. So this is a brief abstract of what exactly I have done throughout my journey of my PhD. First clipping, you can see that it's a petri dish having some brown color, and in between you can see clear zones which had been cleared. So these are the it's a Lugol's iodine assay. What we have done to identify my organism, how much it is able to produce certain products, okay, and how much it is acting on a substrate which is called as inulin. Maybe you may be hearing about a starch, but I'm going to talk to you something about inulin. Starch, you all know, it is one of the plant storage polysaccharide, and the second most abundant plant storage polysaccharide in the plants is called as inulin. Okay, it is very interesting to know about it. So as I move ahead, let me talk to you more about it. So you can see that in the first one, the zone of clearance indicates that one of the microorganism which we have isolated was able to degrade the inulin that has been supplied in the culture media. Okay, so from there we started, and 68 isolates we were able to isolate it, and out of that one, 11 isolate was found to have one enzyme production which is called as inulinase. So we shortlisted one single microorganism which was able to produce high amount of the inulinase enzyme, and that organism we have characterized it by using 16S rRNA sequencing, and the sequence has been deposited in NCBI. It's available. So if you check with the accession number, you'll be able to get the 16S rRNA sequence of the organism from where exactly the study started. After isolating the study microorganism, we went on with the next step where we have optimized the physical parameters. I have the microorganism, but we should give the proper temperature, culture media, carbon, everything, so that my organism will reach and will get the result appropriately. So that's why we went with optimization physical parameter. After doing that one, we have done even the carbon, nitrogen, as well as mineral nutrient. We have done the optimization. For that one, we have used a statistical screening analysis. It's called as bracket Berman design. So we have used the design to clean out the best carbon source and the best nitrogen as well as the best mineral nutrient required for the what we have got it. So interestingly, my organism says I want only dahlia. Dahlia is one of the richest source of inulin, and it says I want yeast because that is one of a good source for B-complex vitamins for nitrogen source. And it said chloride. So we shortlisted for the carbon, nitrogen, as well as mineral nutrients. Using and having the having all the nutrients, we went with the fermentation, and the organism happily grew and it started to produce an enzyme. We initially called it as inulinase. When you get the classification of enzymes, it was found to be as endoinulinase. So that is very important here, because when inulin substrate is acted upon by exoinulinase, you get a product which is the fructose. Whereas when it is acted upon by endoinulinase, you get a product. That's very interesting. That's how research continued further. The product is called as F. It's an oligosaccharide, oligosaccharide. And once I got that product, I started to recreate, because I have to do some application studies. So I have to recover the products. We have used various methods and techniques in order to recover the product. After recovery, you have to characterize it. You should know, yes, only I can tell that it is producing FOS. To prove it, so I have used HPLC, NMR, as well as other techniques in order to characterize the product. After characterizing the product, we went with three different application studies. One is which I'm going to discuss with you now. The effect of the FOS on the probiotic organisms is the organism after feeding on FOS, is it able to produce the postbiotics? So that was the area what we are going to discuss. And even I've studied some other applications of the effect of FOS on the cytotoxicity test using HCT116 cell lines, which is to check whether it's having any impact on the colon cancer cell lines. And we have synthesized nanoparticles with the FOS, and we have studied that also, like characterized it, and we have used it for the in vitro cytotoxicity study. Okay, so this is just a brief outline to make you to understand what exactly and how I was working to complete my PhD work. Okay, so coming to the introduction. So inulin is a polyfructan. Okay, you know starch. So it's made up of just alpha D glucose molecule, very much familiar to us. But the second familiar one, you all get familiarized to it, children. The reason is because your gut needs this inulin. Okay, and you may be wanted to know what are the food sources which are rich in inulin. You're consuming it knowing or unknowingly daily in your food, and one of the richest sources, onion. Okay, you're consuming it, I think. So everybody consumes, many of us consume onion. Garlic is another source. Asparagus is another source. Okay, these are the major sources. Chicory, coffee, Yeah, so it's one of an the compound which we mix along with the coffee. So chicory contains high amount of inulin, 15 to 20% approximately. It is containing. Along with that one, certain other food materials like wheat, barley, rye, they also contain, but the percentage is too less. But the richest source is onion, okay, garlic. And dahlia is there, Jerusalem artichoke. We don't consume Jerusalem artichoke, it's a root tuber. We don't consume it in European countries. It's one of a tuber. They consume it in lot, like how you eat your potato chips. They consume their Jerusalem artichoke chips over there. So like that, we have got all these materials and we have done the experiment. And as I said to you, my organism was preferring only the dahlia inulin. We don't consume dahlia, but my organism demanded that one. Okay. And when I say that inulin, it is an natural reserve material. It is non-digestible. That point is very important, children, because starch you can digest it because it has alpha 1,4 glycosidic linkage, but inulin contains beta 2 to 1 glycosidic linkage, and no enzyme in our biological system can digest it. That's why when you're consuming it from your oral cavity to the digestion, it just goes as such how you have eaten it, and your gut microbe waiting over there for it to undergo metabolism. So those are the organisms which can break the beta 2 to 1 glycosidic linkage. So they are non-absorbable, and inulin contains a repeating unit of the beta D fructose. Okay, and each of this inulin molecule will be ending at the terminal end with alpha D glucose with alpha 1 to 2 glycosidic linkage. But remember, each of this inulin molecule, the chain can go up to 200 residues depending on the plant source what we are going to consume. Okay. So coming degree of polymerization also is increased. That's nothing but the repeating unit of the fructose molecule. And you can see the mode of action of inulin. I just told orally. You can see its mechanism of action over there. So you can find that the FOS is okay. FOS is being acted upon by the exoinulase, and you can see the product is what? Beta D fructose. And beta D fructose also is not very just a normal sugar. It is a sugar with which you are able to produce your biofuel, ethanol. You can produce fructose, high fructose syrup. You have heard about HFCS, high fructose corn syrup, but I am talking about ultra fructose syrup only obtained from the inulin molecule. And now people are looking forward to use inulin as one of an a backbone for the production of the high fructose syrup. The reason is because it's only one single step, and whereas in the case of corn, you have to go ahead with three different enzymes, three different steps, and the last step is very crucial because when you use the glucose isomerase, you will get only 45% of your fructose molecule. But when I use inulin and when I use exoinulase, I will get 95% of the fructose just with one single step. So that's why even this enzyme is very interesting, and many people are working on that one. But my focus was on FOS, so I was trying to isolate the organism, and it gave the enzyme endoinulase, and the product is said to be as fructo oligosaccharides. Yes, are you all with me? Yeah. Okay, so what is interesting about these oligosaccharides as fructooligosaccharides? Point number one, they are going to produce, when they are being fermented, when these fructooligosaccharides are being fermented, they produce bioactive and bioavailable compounds, especially the short chain fatty acids. From morning, maybe you must have come across a lot about SCFA, short chain fatty acids, then B complex vitamins, microbial polysaccharides, and organic solvents like ethanol and bacteriocins. Among these short chain fatty acids, because my area of study was related only to the short chain fatty acids, I'm trying to highlight over there. So when we talk about the short chain fatty acids, three fatty acids, please remember, children, and the first one is the butyric acid that plays a very, very crucial role in our biological system. Then is the propionic acid and acetic acid. Other than that, also you have valeric acid, isobutyric acid, isovaleric acid, various other derivatives are also there. But out of all the short chain fatty acids, the most important one is the three short chain fatty acids what I've given you, and out of that three, the most important one is the butyric acid. So what are their role? What do they do? What exactly these short chain fatty acids does in our body is they are involved in immune modulation. They regulate inflammation in the mucous membranes. They enhance the absorption of mineral ions, prevent colorectal cancer, and also they remove nitrogenous compounds. So these are some of the important roles played by the short chain fatty acids. Now again, you may ask me, suddenly ma'am is jumping from FOS to SEFA. Okay, so I link you and connect you how it happens. So you can watch a small movie, just 1 minute movie, you can watch it. I'm going to tell to you about how the mechanism. I am trying to link prebiotics on probiotics with the postbiotics. So just have a look at it. Point number one. So you can see that it is onion. You have seen, okay? They all are prebiotic foods. So these are all the prebiotic foods which you are consuming regularly in your diet. And then they are, when you're consuming it, as I told you, they are rich in inulin. They are not digested with the oral enzymes. No digestion happens even in your acidic pH of stomach. Okay, directly it reaches into your gut, and gut you have your organisms which are all different types of your probiotic organisms. Okay, what do they do? These probiotic organisms will feed on your inulin or its derivatives, that is oligosaccharide, that I'm talking about fructo oligosaccharide, to produce short chain fatty acids, which includes propionic acid, acetic acid, as well as butyric acid. It also produces various other products, what I told you. What happens now? If they are all acids, your pH in your gut will decrease. Okay, that plays a role where it will inhibit the growth of your pathogenic microorganism. Point number one to be remembered. Okay, and also these products via various different types of the receptors will go into the various other parts of your body, like the gut. In the gut itself, it will have some role. In the brain, it has got a huge crosstalk with your brain. That's why your gut will decide, your gut microbe will decide your mood set, your mindset. Okay, so there is a huge relationship between what you are eating and what you are doing every day, and that is also having an impact on the cardiovascular system and also mineral absorption, bone. Okay, so this is what is a complete understanding and relationship between your prebiotic, what you eat, what your probiotic organism does, and what are the products as postbiotics you get, and how they are impacting your biological system. So this is just an introduction, but my methodology whole thing included two probiotic organism. One is called as the Lactobacillus plantarum, and the other one is Lactobacillus rhamnosus. I've given you the species, and these are the experimental parameters what we have done, and you have the MRS media. Okay, in that one we have cultivated it. What we have done is that in the MRS media we have replaced the carbon source instead of dextrose with my product, that is FOS. So I told you, I have taken an organism, and my study organism was Bacillus teculensis VAK19. So that is been deposited in the NCBI. So we are, by using that organism, it has consumed inulin and it has used endoinulase. By using that enzyme, we were able to recover the product which is called as the fructo oligosaccharide, which we are labeling it as microbial FOS, MFOS. So in order to have a comparative study, we have purchased one FOS from the market, which I've labeled it as commercial FOS. So instead of dextrose in the culture media, we have replaced with the MFOS as well as the CFOS. And see, these are the experimental procedures what we have followed, because I want to do gas chromatography, because short chain fatty acids, you don't have any estimations or colorimetric methods. The best method to identify their presence is either by HPLC or gas chromatography. I went with gas chromatography, and so these are the results what we have got. So first one is to check out how the prebiotic is stimulating the growth of your probiotics. I've chosen two probiotic Lactobacillus species, and we could find that the colony forming units, as well as we can find that there is a huge impact in the growth rate of the microbial microbes what we have used. And if you compare the graph, you can find that In the case of MFOs, different percentage we have given 1%, 2%, 1.5, and 2%. When compared to the commercial, also the lactobacillus was preferring to use the microbial FOS. The same information you can find it even in the second part of your graph, which includes the products colony forming units. How many colony forming units? And the MFOS was showing you a better colony forming units when compared to that of the controls what we have used. And as I told you that when SCFAs are produced, what will happen? pH will decrease, acidic pH. So we have checked the pH of the culture media, and when compared to the control, we could find that there is a drastic change in the pH of the culture media, either in MFOS as well as with the CFOS. So these are the results what we have got after gas chromatography. So whenever you're going with your characterization gas chromatography, you require standards. So as standards, we have used a full to predict it. But I told you that out of the three, the best one is which one? Butyric acid. So when I gave dextrose in the culture media, lactobacillus was preferring it to ferment to give what product? Only the propionic acid. Okay. Now coming to your next graph, you can find that I've supplied with MFOS, that is microbial fructooligosaccharides, which was my product. So when we have checked with them, the lactobacillus was fermenting this product to give you the very important short chain fatty acids, namely the butyric acid. So we have done the calculation and found even the concentrations also. Similarly, the same both the organisms when they were grown on the commercial FOS, they also were able to utilize the FOS to produce the butyric acid only. But the only difference is that MFOS, the concentration of butyric acid was much higher when compared to that of the commercial FOS. Now, doing all these things, one interesting point was I started from my basic isolation of an organism, and I told that it is Bacillus techulensis VAK19. That's the isolate which is able to produce endoinulase enzyme. So we wanted to make a clear understanding: can my organism also produce any short chain fatty acids? Because I gave only time limit of 24 hours for the organism to grow on the Dahlia inulin. So for 24 hours, my organism is able to produce which product? MFOS. Okay, so I extended my incubation time for another 24 hours, and then I took the samples and gave for what production? SCFA production checking. And I found that my organism is utilizing the MFOS, but it is forming acetic acid as its end product in the SCFA. Okay. So these are the conclusions of the last part of my application, what I have done. So the microbial FOS produced has stimulated the growth of the lactobacillus, that is the probiotic strains what we have used for the study, and also Bacillus techulensis. Then production of one of the postbiotic product, that is a short chain fatty acids, which was recovered and analyzed by the gas chromatography. And short chain fatty acid metabolites concentration produced by Lactobacillus plantarum OUBN2 on 2% of MFOS and 2% of CFOS was found to be 44.52 microgram per ml and 51.88 microgram per ml. And as I told you, they both were able to produce butyric acid. And similarly, when I put it even on the Lactobacillus rhamnosus, it also produced butyric acid, but the concentration was slightly different. The results indicate that the prebiotic microbial MFOS has a potential to stimulate which organism? Probiotic organisms, resulting in production of the postbiotics, especially the short chain fatty acid, namely the butyric acid. Thank you. Hope you understood, and any doubts, you are please welcome.
Since Vanita ma'am is there with us, I think you can come back and interact with ma'am anytime and discuss on this topic. Now the next speaker is Dr. S. Shalini Devi, who will be giving us a brief glimpse of plant microbiome. So stay tuned with her. Welcome, Shalini.
A very good afternoon to all the delegates of the dais and to my all dear students. Hope my students don't need any special introduction about me. This is Dr. Shalini. And I have been working from past 10 years with Bhavans Vivekananda College. I have 10 years of teaching and research experience as well. To start with, today I'm going to talk about plant microbiome. The topic of our today's national seminar being microbiome. You all were tuned to only listen about gut microbiome from today morning. But don't forget that plant microbiome also plays a very key in designing gut microbiome of human being. How is that going to happen? We'll see in my lecture. So, the contents that I'm going to brief you all today includes introduction to plant microbiome, why should we nurture plant microbiome, my research contributions in the field of plant microbiome, which would include selection and introduction of fittest plant microbiome, tailoring plant microbiome to overcome current challenges, identification of plant microbiome to mitigate drought stress, plant microbiome to reduce oxidative stress, plant microbiome to induce plant immunity, characterize the pathogens to develop right plant microbiome, elicitors of plant microbiome, which can guide us to develop novel biopesticides, and synthesis of nanonutrients that are compatible with plant microbiome. And the last one is microbial metabolites to control plant diseases. These were the these were not just the side headings, these were the research topics which I have dealt with from past 10 years. Coming to the introduction about plant microbiome. Plant microbiome would include all the microorganisms associated with plant. When I say associated with plant, they can be the microorganisms which are present on the plant, microorganisms associated with the root of the plant, in and around the root, and also endophytic microorganisms which can enter inside the plant cells and help the plant in its growth and immunity. So, this plant microbiome of a particular plant would depend on several factors, which include the type of the soil where the plant is growing, the age of the plant, the environment where exactly the plant is growing, the species of the plant, the age of the plant. All these factors are something which can tailor the plant microbiome associated with a particular plant species. So, this plant microbiome can actually influence the kind of nutrients that can be taken up by the plant, and how exactly the plant is going to utilize the nutrients is also dictated by the plant microbiome. That's why we often called in many articles, we find calling plant microbiome as plant's second genome. This is because the plant microbiome is going to dictate, is going to design the plant health, is going to design the plant immunity and several plant growth factors. Yes, now we spoke so much about friendship between plant microbiome and plant, but why are we interested in this topic? We should be interested in this topic because this plant microbiome is the one which can control the biocontrol activity, I mean, which is going to fight with the plant pathogens. It is the one which can mitigate climatic changes. It is the one which can induce plant immunity. We all take vaccines, right? Is there anyone who is going to give vaccines to plants? It is the plant microbiome which can vaccinate the plant, induce the immunity. It is the plant microbiome which can make the plant to overcome metal toxicity stress. It is the plant microbiome which can help plants to overcome to overcome many other biotic and abiotic stresses, and also it is the one which can help us to deal with eco-friendly agriculture strategies. Yeah, why should we nurture plant microbiome? I want you all to have a look at the right side portion of the slide, which is showing you the endosulfan. Endosulfan is a well-known pesticide. If you are introducing say 10 mg per kg of soil endosulfan, remember that in 1 kg of carrot that you are eating, around 1 mg of endosulfan is coming along with 1 kg of carrot. Similarly, 1 to 3 mg of endosulfan is entering in your food if you are eating 1 kg of potato. Means try to understand that there is a relationship between the pesticides that you are introducing into the soil and the food that you are eating. Now, looking at the left side portion of the slide, we understand that whatever the pesticides we are introducing into the soil is entering into the plant material. From the plant material, it is being entered into the insect gut, because insects are the herbivores which are going to live on the plant material. Then from the plant material, it is also entering the human gut. So what happens if this takes place? If you are not taking enough care of plant microbiome, this is going to affect, this is going to cause higher utilization of pesticides. Means if we are not protecting the plant microbiome, obviously the plant health will come down. To enhance the plant health or to enhance the plant growth, you have to use more amount of fertilizers, more amount of pesticides, chemical fertilizers and chemical pesticides. These chemical pesticides or fertilizers in turn are going to cause evolution of pesticide resistant insects. So there can be gene transfer of resistance genes can enter into different insects and they can enter into ecosystem, which is again a great threat to our environment. And also, as I told you just now, there can be dysbiosis in the human gut, there can be dysbiosis in the animal gut, there can be dysbiosis, I mean production and survival of wrong or bad bacteria in our gut can also happen because of introduction of pesticides. This is the chart which shows the right connection between not protecting, what happens when you don't protect plant microbiome, what happens to our gut microbiome. This is the slide which gives you the overview. So I stress upon protecting the plant microbiome. From here, I will take you through my research contributions in the field of microbiome. To protect the microbiome, what can we all do? As researchers, article where we have studied plant microbiome which can help in combating fusarium related diseases. Fusarium is a notorious plant pathogen which causes root rot disease in potato, in tomato, and especially in citrus plants. It is a notorious plant pathogen. To fight against fusarium, we have isolated 14 native trichoderma isolates. I again called native trichoderma isolates because remember, the plant microbiome which can thrive in a particular environment may not thrive in some other environment. I mean, if I am isolating a plant microbe from maybe Arunachal Pradesh or Himachal Pradesh, that may not work for me in Andhra Pradesh and Telangana because the environmental conditions are quite different. So it is always important to capture, to identify the native isolates, and the native isolates you have to look for which is the fittest among the native isolates so that you can improve and encourage the growth of native and fittest plant microbiome in the soil. Then we can also tailor the plant microbiome to overcome the current challenges. We were all talking about pesticides and fungicides. But we may prevent using pesticides and fungicides maybe from now. But remember that already most or all of the agricultural fields are fully laden with fungicides and pesticides, because we were using all these fungicides or pesticides from years not known to us, right? At least from 100 years we were using these, and we have already filled our agricultural fields with pesticides and fungicides. Now the problem is, when you already have a fungicide in the soil, the fungicide cannot differentiate between the good bacteria or the good organisms and the plant pathogens. You were actually introducing fungicides in order to kill the plant pathogens, but the fungicide is also killing your good plant microbiome. Please try to understand that. To overcome this challenge, what exactly we have done is, we have took the trichoderma isolates, which is again a positive plant microbiome to say, and we have developed trichoderma isolates which can tolerate different levels of fungicide, which is carbendazim, which is a widely used fungicide. And now this trichoderma isolate has been commercialized in various cities in Andhra Pradesh. This product is actually available, a mutant strain of trichoderma. So in this way, you can tailor, you can orchestra the microbiome according to the need, according to the growing climatic changes. Then, identification of plant microbiome, fight with drought. This is the work which I have done in Telangana along with one of my PG student. So we know that many areas in Telangana are drought prone. So you can again nurture, you can again identify plant microbiome which can help plant to mitigate drought stress. We have identified 3 bacillus isolates which can help plant, even if you are not watering the plant maybe for 10 to 15 days, and if you are giving drought conditions to plant, still the plant can survive when you inoculate this bacillus bacteria into the soil. So to fight with drought also, again, you can take the help of plant microbiome. Then you can also use plant microbiome to overcome heavy metal toxicity. Under areas where there is a lot of industrialization, the soils are filled with toxic levels of lead, where again the plants can undergo oxidative stress. To overcome oxidative stress by helianthus plants, we have again tried trichoderma isolates which can help the plants to overcome oxidative stress. This is a paper published in Indian Journal of Experimental Biology. I think the year is 2017. Then the important contribution of plant microbiome is induction of plant immunity. As I told you already, we all took vaccines when corona has come up, but who is going to vaccinate the plants? Everybody think of spraying something onto the plant only when the plant is diseased, or when your plant is drying up or affected, you think of spraying some pesticide, some kind of insecticide on the plant. But why don't we prevent that plant disease? Prevention of plant disease can be achieved by induction of plant immunity, which can be again done by nurturing right plant microbiome. I have isolated streptomyces isolate, Streptomyces albolongus, in fact, which is known to induce plant immunity by secreting a variety of fatty acids, which includes hexadecanoic acid, lanolenic acid, and other HA acid, etc., which is hypothesized to enter the plant cell, to enter the plant cell wall, and it undergoes a series of metabolic reactions, and finally that is going to trigger the production of salicylic acid and jasmonic acid. Salicylic acid and jasmonic acid are important in fighting with plant pathogens. They are important in induction of plant diseases. This is the hypothesized metabolic pathway, which we have presented in a paper published during 2018 in the journal called Environmental Sustainability. So, moving on to the 6th weapon which we can use to protect plant microbiome or to nurture plant microbiome is identify the enemy before you fight with them. right identification of the enemy is very much required. So we have also, I have collaborated with scientists at Mysore CSTRI, where they were working on identification of mulberry disease causing pathogens, mulberry root rot causing pathogens. We identified Lacediplodia theobrome isolates, which was the first report to be reported in case of mulberry plants. And we could isolate and identify this, so that we were now working on what is the right plant microbiome. We were working on designing the right plant microbiome to combat the infections of this Lacediplodia theobrome. Then elicitors from plant microbiome, we have to learn from plant microbiome. So if we know what exactly they were trying to do to plants, it will be easy for us to screen many novel biopesticides. So this is a bioinformatics work where we have compared various sequences of the elicitors of plant microbiome, so that we may design novel elicitors. It will be easy for us to design novel elicitors to induce plant immunity. Yeah, this is something, a work which is not yet published. Whenever we talk about nanoparticles, everybody nowadays we know about nanoparticles, and the buzzword is nanoparticles. When we talk nanoparticles in relation to microbiology, we always say nanoparticles are something which can inhibit microorganisms, which shows antibacterial activity, which shows antifungal activity. We have n number of research publications talking about this. But whenever these publications were seen by me, I was with a question that also I want to tell you that many companies were synthesizing nanonutrients for plant growth promotion. Nanonutrients on one hand, nanoparticles showing inhibitory activity against beneficial microorganisms on the other hand. How to go with it? So we want the plant microbiome to be sustained. We want the nanonutrients to help the plant growth. But obviously, nanoparticles are always antagonizing or always inhibiting the microbial growth. So in the top pictures, you can see manganese nanoparticles and cobalt nanoparticles, which were promoting seed germination, seed vigor, and also the plant growth. Then we have along with the cooperation of our physics department, BVC, we have synthesized nanoparticles which were compatible with plant microbiome. In the bottom slide, you can see that few areas were not inhibited. Few areas were inhibited by nanoparticles, and few regions were not inhibited by nanoparticles, where there is good growth, luxuriant growth of trichoderma. Trichoderma is again one best known plant beneficial fungi. So here we report the synthesis of nanoparticles, I mean nanonutrients, which have compatibility with plant microbiome, so that you can introduce both plant microbiome along with nanonutrients into the soil. We are ensuring that the nanoparticles are not killing the plant microbiome. They are in fact safeguarding the plant microbiome. So maybe such nanonutrients has to be commercialized. So coming to microbial metabolites, plant microbiome do synthesize several array of metabolites which can antagonize the plant pathogens. So instead of going for pesticides, instead of going for chemical fungicides, if we can identify the metabolites from the plant microbiome which can inhibit plant pathogens, which can promote plant health, we may synthesize these microbial metabolites chemically, so that these microbial metabolites, since they are of natural origin, they can be degraded naturally in the ecosystem. So we have identified two compounds. One is octadecanoic acid and the second one is a derivative of natamycin. These two compounds were extracted from Streptomyces albulongus. Streptomyces albulongus, which is again a part and parcel of plant microbiome. And these compounds can be used to induce plant immunity as well to inhibit the pathogens. In the picture shown over there, you can see the inhibition of Cornospora cassicola, which is again a notorious plant pathogenic fungi being inhibited by the microbial metabolites by Streptomyces metabolites inhibiting Cornospora cassicola. So I would like to conclude with one of my review papers. So far, what all I spoke is all about the 9 papers where my research articles. This is my review paper with which I would like to conclude saying that if you protect the microbial wealth, that is going to protect the plant health, microbial wealth for plant health. If you protect the plant health, obviously, as we discussed in the starting, that is going to protect our gut health. Thank you so much for providing me this opportunity. My heartful thanks to Dr. K. Anuradha, Head Department of Microbiology, for giving me this opportunity. My heartful thanks to management of Bhavans Vivekananda College for always supporting me in all the research and teaching activities. Thank you a lot to my all dear audience. Even if it is this is lunchtime and still I think you were all focused to my lecture. Thank you so much.
Thank you, Shalini ma'am. Since please, sir.
Yes, please, sir.
Just wanted to have one question. Plant microbiome, you're re-giving it to the plant? You extract plant microbiome and then again giving it to the plant for protection or the growth?
Yes, sir. Actually, let me tell you.
Or you're developing that into some kind of a chemical and then giving?
I spoke about both the things, sir. In the first 2 articles, whatever I have spoke, we were trying to isolate it from one plant source or from one soil and introducing it into other soils. We are trying to isolate the best one, fit one, so that even if there are environmental changes, climatic changes, this would thrive.
So does it happen? Depend. Anyway, soil difference is there, but does it impact if the plant is different? Means one plant micro can it give help to other kind of plant growth
sir.
also?
Yes.
It's not dependent on the same plant. That means if you have to reproduce the same plant once you treat, or we can go different plants.
We can go for different plants, sir. Actually,
so it is impact is there on different also. It is.
Yes, sir. Yes.
Just that one clarification.
We also checked with different plants. That work is going on with some of my UG students right now going on, but still. Even according to literature also, we isolate it from one rhizospheric soil and we can introduce it into many other soil types can be introduced.
So there you can go any type of plant. There is no restriction.
There is no restriction.
Like for example, if sugar cane is grown, you reintroduce there, it should be only sugarcane plant or it can be some other plant?
No, it can be any other plant.
Yes,
yes, it can be any other plant.
Okay. Thank you.
Any more questions? Yes, sir.
See, as a layman,
yes, sir.
I would like to have one clarification. Long back, many of agriculture lands were converted into prawn culture lands, where salt water is induced. And now we are talking about this plant, these things. So what will happen to such kind of lands? Can we convert them again into agriculture lands, and can we do normal agriculture in that?
Yes, sir, we can. This can happen, as I told you already. We have to select which plant microbiome, I mean, which microorganisms can thrive in that particular soil, sir. Because some microorganisms can tolerate high salt concentrations. Uh, right now, we were working on one species of microorganism which can thrive at 7% salt concentration. So 7 grams in 100 ml is 7%. Even in 7% salt concentration, that organism is thriving. It is also promoting the plant growth. So if we can do research in identifying such plant microbiome, I mean such microorganisms can be introduced even into high salt contaminated areas can be introduced, sir.
Thank you.
Thank you so much.
Thank you, Shalini. That was a real enlightenment about the plant microbiome. So all this while everybody was talking about human microbiome, and now we are actually talking about One Health concept. Basically, the One Health concept revolves around all the types of microorganisms flourishing everywhere around us. So now, after these enriching sessions, now we move on to the lunch break. There will be, I invite the dignitaries to join us for the lunch, and also the students. I request all the students to kindly join for the lunch with the food coupons. Let me say that the sessions are a little delayed, so therefore we have restricted the time to 40 minutes. So kindly join again for the next session sharp after 40 minutes. Thank you. Food coupons are mandatory. So kindly take your food coupons and then join for lunch. All the participants, those who are going to do oral presentations, those who are offline are requested to give their PPTs to me, PowerPoint presentations to me after lunch.
ये सब अभी टाइम ये सब चल रहा है ఇది ఆన్లైన్ Manjusha. I think after Madhurima, Manjusha, you can your lecture. Then Kushbuji. Online class పెట్టుకుందాం. First offline ని చేసేసి. Okay. First we'll finish up offline and then we'll go for online. It will be continuation, right? So I think first Madhurima and then Manjusha. I think after Madhurima, Manjusha, you can your lecture. Then Kushbuji. Online class పెట్టుకుందాం. Yeah, free. Okay. First we'll finish up offline and then
Hello, good afternoon. I request all of you to settle down. We'll start our oral session.
and then Manjusha. I think
We are having 6 lectures now. Out of 6, 3 will be offline and the remaining will be online. Okay? And I request the participants. Hello, good afternoon. I request all of you to settle down. We'll start our oral session.
and then Manjusha. I think
We are having 6 lectures now. Out of 6, 3 will be offline and the remaining will be online.
Dr. A. Madhuri from Government Degree College.
Good afternoon. I request all of you to settle down. We'll start our oral session. We are having 6 lectures now. 6, 3 will be offline and the remaining will be online.
Dr. A. Madhuri from Government Degree College.
Good afternoon. I request all of you to settle down. We'll start our oral session. We are having 6 lectures now. 6, 3 will be offline and the remaining will be online.
Dr. A. Madhuri from Government Degree College.
So my presentation is about using agricultural waste, that is green gram for enzyme production using solid state fermentation. Myself, Dr. A. Madhuri, Assistant Professor of Microbiology. So my presentation is about using agricultural waste, that is green gram for enzyme production using solid state fermentation. Myself, Dr. A. Madhuri, Assistant Professor of Microbiology. Good afternoon, one and all. Myself, Dr. A. Madhuri, Assistant Professor of Microbiology from Government Degree College for Women Microbiome. My today's presentation is about using of green gram portions as a substrate in solid state fermentation for production of alkaline proteins. Good afternoon, one and all. Myself, Dr. A. Madhuri, Assistant Professor of Microbiology from Government Degree College for Women I have done under supervision of my guide Gopal research is about as we know what are green grams and enzymes as we substrate in breakdown fermentation and effective are these are active under analyze pH. And my work is focusing on using of agricultural waste as a substitution of fermentation. And I have used many substrates of this green gram portion. And as you know, this is one of the main products in India. And after the production, the portions will be removed as some is used as a noise, and the remaining will be going as a waste. So this substrate is used adapted for using as a for production of enzyme in this world. As you know, this is one of the main products in India. And after the production, the portions will be removed as some is used as a noise, and the remaining will be going as a waste. So this substrate is used adapted for using as a for production of enzyme in this world. You know, this is one of the main products in India. And after the production, the portions will be removed as some is used. In this work, we have used organism as well as this is the aspect of the lab. And we have used solid state fermentation and substrate as we have discussed. I have used green gram portion. And for this substrate, we have taken different particle sizes. That is less than 1 mm, 1 to 2 mm, 2 to 3 mm, and more than 3 mm. fibers. And for substrate we have, as we are doing new solid state fermentation, we have checked machine and and in for this substrate we have taken different particles in the formula for calculating moisture content of the substrate. First we have to dry the substrate till that takes a constant weight, and then we have to calculate the moisture content of the substrate. And this is the medium we have used a solid state fermentation, that is medium solid medium, and this is the composition. And particles in the formula we have 2 ml of moisture culture substrate. First we have to dry the starch till that takes a constant weight, and we have used parallel sizes, that is the medium, and for enzyme extraction we have used glycogen buffer, and it can be roasting for 1 hour. First we have an extra starch till that takes a constant weight, and we have used parallel sizes, that is the medium, and for enzyme extraction we have used glycogen buffer, and it can be fermentation, we have to extract the enzyme, and for that enzyme, we have to do alkaline protease assay to know how much enzyme has been produced. For that, we have to take 0.5 ml enzyme and we have to mix it with the substrate, because enzyme has to use some substrate. That is casein. Here in dissolved in buffer, glycine NaOH buffer, and we have to allow the reaction to happen for 30 minutes, and after 30 minutes it will be terminated by TCA and centrifugation will be done. And after that, I have added 5 ml of 0.5 molar sodium carbonate and 0.5 ml of 1 molar phenol reagent. It is allowed for 20 minutes, and then observance can be read at 60 nanometers against tyrosine spectrophotometer. And 1 unit of alkaline protease produced in this method is measured as 1 microgram tyrosine per minute under standard assay conditions, and it is taken as protease activity per gram dry substrate. 1 unit of alkaline protease produced in this method is measured as 1 microgram. Of the fermentation, we have analyzed the substrate, whether the nutritional content of the substrate has been increased or not. For that, we have checked ash content, fruit fiber, protein, carbohydrates. Of the fermentation, we have analyzed the substrate, whether the nutritional content of the substrate has been increased or not. For that, we have checked fruit fiber, protein. We have seen we have taken four different particle sizes, that is less than 1 mm, 1 to 2 mm, 3 mm, and more than 3 mm. And moisture content, we have taken 5%, 60%, 70%, and 80%. And incubation period, 24, 48, 72, and 96 hours. So we have observed highest enzyme production and with particle size 1 to 2 mm. We have taken 5%, 60%, 70%, and 80%. And incubation period, and optimum incubation period is 48 hours, and moisture content is highest 70%. After getting these optimum conditions, we have checked for effect of carbon sources and nitrogen sources, and optimum incubation period is. We have added different carbon sources like glucose, maltose, lactose, xylose, and starch. But we have seen the reduction in enzyme production with all the carbon sources. And coming to nitrogen sources, we could say improved production. All the nitrogen sources except soya mustard have shown increase in production, and the highest enzyme production was achieved with ammonium sulfate. Under all these nitrogen have checked whatever substrate is left over after fermentation. We have checked for nitrogen sources except soya mustard have shown increase in production, and the highest enzyme production was achieved with ammonium sulfate. Under all these nitrogen have checked whatever substrate is left over after fermentation. We have checked for nitrogen sources except soya mustard have shown increase in production, and the highest enzyme production was achieved with ammonium. After fermentation, we could see difference in substrate, that is protein content
PhD under supervision of Dr. Hamidabi. And okay, coming to my topic, this is under probiotics, prebiotics, and postbiotics. And title of my today's paper will be the insights into articulate yeast Hanseníaspora species isolated from the fermented beverages. Actually, when you think about yeast, then I am sure that you will be imagining about Saccharomyces cerevisiae, Saccharomyces boulardii, which are probiotics into articulate, and according to the grass studies also isolated from. And apart from Saccharomyces, you will be having n number of yeast, for example, Candida, Microbiome, and Hanseníaspora are easily explored. If anybody wants to do the research, then please focus on the rest of the yeast, not only the Saccharomyces. Saccharomyces, you will be having n number of yeast, for example, Candida. Coming to the introduction, Hanseníaspora is a genus of yeast, and it also part of a fungi, Ascomycota phylum, and subphylum Saccharomycotina. Saccharomyces is class, order will be Saccharomyces. Coming to the family is Saccharomycoidaceae. Hanseníaspora being considered in the Saccharomyces, but it is known as non-Saccharomyces. It is also part of according to today's classification. And Hanseníaspora, when it is endomorph form, it is also known as Floacara. Coming to the family is Saccharomycoidaceae. But the microorganism is the same Saccharomyces terminology is known as non-Saccharomyces. They are typically reticulate endomorph form. It is also known as Floacara. Typically they happen in greater pre-fermentation, and the first terminology is known as non-Saccharomyces. They are typically in 1842 to 1909, who was a Danish mycologist and fermentation psychologist. Actually, he is isolated from the orange. It's a very new thing because all of you think that orange is some acidic. It's not isolated. So it is a myth. Okay, because from orange he has isolated this particular Hanseníaspora genus in pre-fermentation. The cell is reproduction by budding. The same mechanism like Saccharomyces it will do. In 1842 to 1909, who was a Danish mycologist and fermentation psychologist. Actually, he is isolated from the orange. It's a very new thing because all of you think that orange is some acidic. In 1842 to 1909, who was a Danish mycologist and fermentation psychologist. Actually, he is isolated from the orange. It's a very new thing because all of you think that orange is some acidic. In 1842 to 1909, who was a Danish mycologist and fermentation psychologist. Actually, he is isolated from the orange. It's a very new thing because all of you think that orange is some acidic. After DNA isolation, we find out that we got Hanseníaspora species on fermented fruit juices. We have constructed phylogenetic tree. In 1842 to 1909, who was a Danish mycologist and fermentation psychologist. Actually, he is isolated from the orange. It's a very new thing because all of you think that orange is some acidic. After that, we have to add a especially for our active culture of your yeast. After DNA into the juices and keep it at the incubate shaker at 30 degrees centigrade for 24 hours generally, not for 48 hours. 24 hours and after that, the Danish mycologist and fermentation psychologist. Actually, he is isolated from the orange. It's a very new thing because all of you think that orange is some acidic. After that, the selected is fermented pineapple juice, fermented banana juice, and fermented sweet lime. These are the three juice shakes selected for the isolation of Hanseníaspora species, not for 48 hours. In 1842 to 1909,
by using color mechanism, we have done the zero-point analysis. And among the pineapple, you can color parallel here. Q angle is H by using color that has shown we have done approximately near another 90 degrees. And among the by using enzymes for a species, its color doesn't get changed. It will look like natural only. It will retain the aroma of the by using and bioactive color component is fermented fruit juices, that is like it will retain the pineapple and plum by apple and bioactive color component is fermented fruit juices, that is and you can see that pineapple you can see high amount and plum phenol, flavonoid, ascorbic acid. The sugar level got reduced. It's very similar. You can see the sugar level pineapple and this is the antioxidant activity. Antioxidant activity, natural flavonoids have antioxidant activity. The sugar level doesn't affect those antioxidant activity. Okay, how it will normally the natural antioxidant activity will be there. Same activity retained. This is the DCMS analysis of fermented pineapple because normally then I have given only the one data, the pineapple, because we have got a high amount of phenolic and flavonoid, so that's why I have taken this particular DCMS analysis further the DCMS. DCMS, you can see the compound name Phenolo. Actually, it will give you a, according to the literature, it will give you a lemon type of flavor. Citronello, citric acid, see the compound name and Veraniol, it will give you some rose type of fragrance. Trans-2-hexacetic citric acid and 1-octanol citrus. I see eugenol. I think Veraniol, you have studied about the flow. This is the main factor which will main compound which will be seen by the octanol. I see eugenol, spicy flow. I think sweet fragrance, the main factor because alcohol, I think you all of you know that it will give you a fruity order. Okay, I see eugenol, spicy flow. I think conclusion is nothing but there are not many studies. Actually, articles I have there are not many studies that are reported on Hexania fluorophore body, especially when compared to the probiotic strains like Actually, I forgot to write Saccharomyces boulardii also. Articles I have there are not many studies. Further, co-protein studies of metabolic pathways present, especially when compared to the probiotic and extraction of nucleic acids and biosurfactants. The enzymes will be able to produce the biosurfactants or not. That is the main pathways of my research. And biosurfactant amino and cosmetic, I think anti-aging creams already much more interesting. So we are looking into that now. And biosurfactant no, if it got a customer in country, acknowledgements, actually, I'm very thankful to the UGC, Sahiba Jyotira Phule, which I got. And coming to the references, acknowledge there is only the one article which has been published till now on maintenance for pseudophilus mondi, which improves the flavor of tilapia fish protein hydrolysis. And coming to the references, using whey proteins, have you heard of the whey proteins which make raw only, like which will give you some bitter taste. Maintenance for a filler mondi will reduce the particular bitterness. Whey proteins, I've seen that the paper, the whey proteins which make raw only. Maintenance for a pseudophilus mondi plus lactic bacillus fermentum or bacteria is there. In that can reverse those particular combinations and then check on the maintenance for aromatic profile, whether you use enhancing aromatic and reducing the bitterness or not. They have checked those particular parameters. And like aromatic profile, only the maintenance for has the potential to reduce the bitterness that protein hydrolysis, not the combination. Lactic acid actually also has to give that much of efficiency compared to the hexanase protein hydrolysis. And this is the microbial art we have done when we have time, and that is the hexanase for that gives that much of efficiency compared to the hexanase. Thank you. Protein hydrolysis.
Thank you, Manjusha.
This is the microbial art.
All the next speakers. Thank you, Manjusha. All the next speakers.
Fatima. They're from University College, Mexico. Now, from now onwards, they'll be presenting the presentation. Next speaker will be Simran and Hazira Fatima. They're from University College, Mexico. Now, from now onwards, they'll be presenting the presentation. Next speaker will be Simran and Hazira Fatima. They're from University College, Mexico. Now, from now onwards, they'll be presenting the presentation. Next speaker will be Simran and Hazira Fatima. They're from University College, Mexico. Now, from now onwards, they'll be presenting the presentation. Next speaker will be Simran and Hazira Fatima. They're from University College, Mexico.
I hope my screen is visible.
Next speaker will be Simran and Hazira Fatima. They're from University College, Mexico.
I hope my screen is visible.
Next speaker will be Simran and Hazira Fatima. They're from University College, Mexico.
I hope my screen is visible. Good afternoon, everybody.
Yeah, I invite Hazira Fatima and Simran to present their talk.
Good afternoon, everybody. Good afternoon, everybody. My name is Simran, a postgraduate MSc Biotechnology student from University College for Women 14. And the title of my research work is Isolation and Evaluation of Endophytes from Hibiscus tubatus for Postgraduate Growth Promotion. This research work was carried out under the guidance of Dr. Hamidati and Dr. Saida Amna Kaushal as a part of graduation of endophytes. I am also working on an extension of this project. This research work was carried out under the guidance of Dr. Hamidati. My research work focuses on exploring endophytic bacteria from Hibiscus tubatus. Endophytes are microorganisms that reside within the plant tissue and are inherent components of plant. Research work focuses on exploring such plant bacteria from Hibiscus.
Simran, 1 second, can you put on your mic? Camera, put on your camera on. Simran, 1 second, can you put on your mic? Camera, now your camera on. Simran, 1 second, can you put on your mic? Yeah, continue. Now your
Plant-associated microorganisms that are known to influence plant growth are broadly categorized under the term plant growth promoting bacteria or they can enhance plant growth by various direct and indirect mechanisms such as nitrogen fixation, nutrient mobilization, and protecting the plant against attacks by various phytopathogens. These plant growth promoting bacteria can be chemically explored as an alternative to chemical fertilizer. In this study, Hibiscus tubatus was chosen as a subject to isolate endophytes as alternative plants of this have wide medicinal properties, and earlier also species have been isolated from Hibiscus rosacea, but this particular species, that is Hibiscus tubatus, is very less explored. Earlier also the three main objectives of this study were: one, to isolate endophytic bacteria from Hibiscus tubatus. The second objective was to screen the isolated bacteria for the three main objectives of this study. And the third objective was to evaluate the best performing strains for transverse promoting activity on and formulate screen the isolated bacteria for the material method and the plant sampling. The third objective was the campus of University College of Women and submitted to Botanical Survey of India as a herbarium for identification. The plant sample was then processed to isolate endophytes. The isolated bacteria was screened for plant growth promoting botanical survey of various tests, and the best performing strains were evaluated by testing for seed germination and seed trigger index of botanical promoting botanical survey of various tests. So the following tests were performed in order to screen the bacteria for plant growth promotion by testing zinc solubilization and phosphate solubilization in order to assess their ability to mobilize insoluble minerals like insoluble minerals, making them more available for zinc solubilization and plant solubilization in order to ammonia and IA mobilize. IA is a phytosoluble that helps in plant growth, making them more available urease activity. A is pH to basic, changes the color to pink. And cellulase activity, appearance of a halotone or one of nearly HCl the positive result, nearly 14 isolates the positive for basic. Lipase activity, changes color of media from pink to yellow in cellulase positive result, almost all isolates the positive for nearly HCl the positive result, nearly 14 isolates. And chemical assay, basic. Lipase activity, 4 isolates, SLR1 yellow in cellulase positive, R9 and L significantly inhibited the growth of both phytopathogenic fungi, and percentage assay was calculated and are given as the graph. Here is table of observation of results for all PPP tests, and after observing the isolates were selected as the best performing strains. The 3 isolates were HLR1, SLR9 and SLR6 for all PPP tests, and these 3 most valuable strains were studied as polymerase. After 7 years of observing HLR1, the index was calculated and percentage germination was determined. HLR9 showed highest chance of promotion during an SCI index at a range of 56. After 7 years was significantly greater than that of control, that showed 3 results. And percentage germination was also higher, that was 50%. The other 2 isolates also significantly greater than control, giving higher SCI index and percentage germination when compared to control. Was also higher, that was 50%. Summary and conclusion, it can be concluded that hibiscus lobatus is a potential source for isolating plant growth from all these bacteria. However, further studies are required to assess their full potential as bioinorganic. Concluded that help to be assessed further by greenhouse studies, etc.
shade and then you shade screen. Yeah. You have an upward arrow icon, right? You have to click on the shade, click on the shade, and then keep your PPT open. Shade screen. Yeah. You have an upward arrow icon, right? You have to click on the shade, click on the shade, and then keep your PPT open. Shade screen. Okay, we will move on to the next speaker. By then you can make any answers. We will call the next speaker. No, we'll give you time at the end, right? Like the other speaker, by then you can make any answers. Okay, next we'll call Srilata. She's also an so we'll give you time at the end. Microbiome department, she'll be presenting her paper now. Okay, next we'll call Taslim. You can log off and then you can join later. So we'll give you time at the end. Microbiome department, she'll be presenting her paper now. Okay, next we'll call Taslim. You can log off and then you can join later. Srilata, put on your camera. Yeah, you're not visible. Put on your video. Taslim, you can log off and back up. Srilata, put on your camera. Yeah, you're not visible. Put on your video. Srilata, adjust your camera so that your face is visible. We can see only your forehead. Yeah. Thank you. Now you are visible properly. Srilata, adjust your camera so that your face is visible. We can see only your forehead. Thank you. Now you are visible. Coming to the introduction part of the Srilata, adjust your camera. Is a homemade polysaccharide composed of fructose, we can see only your basic Levon is produced from homemade sucrose sugar to sources many microorganisms. It could be of a plant or a microorganisms. So by utilizing the sucrose as a source of the plant, Levon the enzyme Levon sucrose sugar classified as other content and other Levon sucrose of a plant or transferase as the bio belongs to the family of glycose of the plant. Levon enzyme Levon sucrose as an enzyme polysaccharide is extracellular environment in the form of the soft amorphous lines. So microbial Levon has been referred as a dietary polysaccharide due to its multi-dimensional applications in the extracellular environment, as well as also anti-cancer and medicinal applications in the extracellular. So coming to the other applications, also anti-cancer and even the structure of the Levon can be of two types. One is of branched type of the Levon and other is linear form of the structure can be seen, and the Levon has been produced both in the plants as well as also by microorganisms. So branched form of the Levon is more of the Levon, and the branched form of the Levon is being seen mostly in the bacteria, whereas the linear form of the Levon is seen in the plants. So here the structure of the Levon can be of two types. One is of branched type of the Levon and other is linear form of the structure can be seen, and the Levon has been produced both in the plants as well as also by microorganisms. So the branched form of the Levon is more. So whereas in the linear form of the Levon and the branched form of the Levon is being seen mostly in the bacteria, whereas the so here the structure of the Levon can be of two types. One is of branched type of the Levon and other is linear form of the structure can be seen, and the Levon has been produced both in the plants as well as also by microorganisms. So the branched form of the Levon is more. So whereas in the linear form of the Levon and the branched form of the Levon is being seen mostly in the bacteria, whereas so here the structure of the Levon can be of two types. One is of branched type of the Levon and other is linear form of the structure can be seen, and the Levon has been produced both in the plants as well as also by microorganisms. So the branched form of the Levon is more. So whereas the Levon and the branched form of the Levon is being seen mostly in the bacteria, whereas so here the structure of the Levon can be of two types. One is of branched type of the Levon and other is linear form of the structure can be seen, and the Levon has been produced both in the plants as well as also by microorganisms. So the branched form of the Levon is more. So whereas the Levon and the branched form of the Levon is being seen mostly in the bacteria,
pharmaceuticals are